this week New England Journal of Medicine on May 9, 2024. In the four original articles, three articles were related to the hematology. Now, first article is hexagamglogin autotem cell for severe sickle cell disease. Second article is hexagamglogin autotem cell for transfusion dependent beta thalassemia. And the third one is the use of recombinant ADAMTS13 for immune thrombotic thrombocytopenic purpura. Hexagamglogin autumn T cell is a basically non-viral cell therapy, and here they reactivate the fetal hemoglobin synthesis by CRISPR-Cas9 gene editing of autologous CD34 positive hematopoietic stem cell and progenitor cells. Now, the CRISPR-Cas9, this is the gene editing. There is another that is called the gene therapy. We will talk about that. What is gene therapy? So CRISPR-Cas9 is a gene editing technique, and it stands for clustered, regularly interspaced, short, palindromic repeats /Cas9. In 2023, FDA approved two autologous cell therapies for the sickle cell. Basically, both of these therapies are curative. One is the lovo-TB-glogin autotem cell. This is gene therapy, and the second one is the hexagamglogin autotem cell. This is the gene editing therapy. Now, gene therapy means introducing a new gene, and gene editing means altering the sequence of the endogenous gene. These strategies modify person's own hematopoietic stem cell, so there is no concern for graft versus host disease and also immunosuppression. So they harvest the autologous stem cell, and they treat the autologous stem cell in the laboratory. And once we have the harvested, treated autologous stem cell, then what we do, we give busulfan to the patient. The purpose of the busulfan is basically a high dose of chemotherapy that kills the cells in the bone marrow. And once we have given busulfan, it's called the myeloablation. Then we re-infuse the harvested and treated autologous stem cell back to the patient. So I was looking for why they use busulfan for myeloablation. Why they don't use any other agent? Why they don't use melphalan? For example, melphalan is used in the autologous hematopoietic stem cell in multiple myeloma. So, but I could not find like the exact answer. Maybe I will further go into detail, further dig into it. But the superficial things I got is this, that busulfan combined with immunosuppression allows efficient engravement of the gene modified cells. In rhesus mac model, this was one study that gave me some idea. In another study, I found that busulfan containing regimens, they significantly spares lymphocyte and maintains a superior immune response to mucosal challenge with simian/human immunovirus, and compared to total body irradiation and melphalan regimens. They found it in the macaque experiments, and this study was done by Jason Murray. So the question I was looking for, why they use busulfan, why not they use the melphalan? So these are the two studies that helped me something, that they had a good response in experiments with busulfan. So that's the reason they used it. But still, in multiple myeloma, we use melphalan 200. That is a famous, it's called the MEL 200. So in lovo-TB-glogin autotem cell, the purpose is to add a functional beta globin gene to patient's own hematopoietic stem cell using the gene therapy through ex vivo lentiviral vector. This will produce more hemoglobin A, so less vaso-occlusive crisis will happen. Now let's talk about the CRISPR-Cas9, which is the gene editing technique. CRISPR, clustered, regularly interspaced, short palindromic repeats. CRISPR is basically the defense technique of the bacteria that they used against the viruses, bacteriophage. Now we know that there are some viruses, they attack the bacteria, they go inside the bacteria, they incorporate these viruses that are called the bacteriophage, they incorporate their DNA in the bacterial DNA, and the viruses, they aim to produce more viruses, like more bacteriophage. Now bacteria, they develop resistance, they develop defense mechanisms through this process of CRISPR against these viruses. So when every time the bacteria, they encounter a new bacteriophage, they cut that small piece of DNA from the bacteriophage and they store into their own genome. Now that stored piece of DNA in the genome will be used by the bacteria in the future if the bacteria comes in the attack of the same type of bacteriophage again in the future. So that stored piece of DNA would help to destroy the attacker. Let's go into a little bit more details. So the DNA from the invading bacteriophage is stored in the bacterial genome at a place called the CRISPR locus. Okay, now the bacteria is ready for the defense. Now let's say after some time, or we don't know, after the infinite time, there is a bacteriophage invasion, there is an attack, there is an intruder coming in. Now we have the DNA from this specific bacteriophage already stored in the genome. Now what will happen? So the bacteria will produce a thing called the CRISPR RNA. It will make a small piece of RNA that is called the CRISPR RNA. Now the CRISPR RNA binds to a large protein that is called the Cas9. Here we use a large protein. Basically the protein is called effector complex, but in our system CRISPR Cas9, we use that effector complex, the large protein is called the Cas9, CRISPR associated protein 9. The purpose of this large protein Cas9 effector complex is to navigate the RNA into the bacteriophage DNA, and their RNA bind with the bacteriophage DNA via same Watson-Crick base pairing, and now the invading bacteriophage DNA is cut, and then again this bacteriophage DNA can be stored into the CRISPR locus for future invasion. So this is the way the bacteria used the information in the form of DNA stored in this genome to protect itself against the bacteriophage and make and to store more information in them, in the bacteria, to prevent themselves from the future invasion from the bacteriophage. Now here we want here that certain genes to be deleted. Certainly, of course, the bad genes, we want the bad genes to be deleted. Now what we need here to do that, to do our job, we need a guide RNA and the Cas9 protein. So the guide RNA will attach with the target DNA, because the guide RNA will recognize the target DNA and will attach there. And because the guide RNA is, we already have a effector complex, guide RNA and Cas9 complex. So the Cas9 complex is an endonuclease, and then it will break the DNA. So once the DNA is broken, then what will happen? The DNA will try to repair itself, but in the process of repair, there are often loss of function mutations. And because of the loss of function mutation, there we will have certain genes will be deleted, and this is what our target is. So this is how, in a simplified and the most easiest way I could understand, I explained you how the CRISPR-Cas9 system works. Now with the help of CRISPR-Cas9 system, we basically block the BCL11A gene. BCL11A gene. So why BCL11A gene we block in sickle cell? BCL11A gene is a switch that controls the conversion of gamma globin to beta globin. So if BCL11A gene is working, gamma globin is converted into beta globin. So what happens? The hemoglobin F is hemoglobin F production starts declining, and more hemoglobin A is produced. Now we want to shut off this BCL11A gene. So when we shut off BCL11A gene, we will be having the more hemoglobin F gene. So this is what will help our patient in sickle cell crisis. We want more hemoglobin F gene, so we shut off the BCL11A gene here. Now the same principle works for beta thalassemia. We have to shut off the BCL11A gene. We shut off the BCL11A gene, we increase the hemoglobin F production. Now this shutting off BCL11A gene business is called the gene editing. Gene editing through the way of CRISPR-Cas9 system. Now we also have the gene therapy, just like we have the gene therapy available for sickle cell. The gene therapy for sickle cell is called the lovo-tibiglogene. Similarly, we also have a gene therapy available for beta thalassemia. That is called the Betti-Beglogene autotem cell. Betti-Beglogene autotem cell.